Everyone. In this class, we'll try to learn about the structure and functions of neurons. The objectives of this class would be to draw a label diagram of neuron, classify types of axons and give example for each, appreciate the concept of axonal transports, their types and functions, list the differences between myelinated and unmyelinated axons, enumerate the nerve growth factors, classify neurons and give examples for each type of neuron, name different type of neuroglia and mention their functions. Neuron is a structural and functional unit of the nervous system. As we know, its primary function is to receive the various stimuli and transmit the signals to other neurons and tissues. An excitable cell where message transmission occurs in the form of action potential. About 10 raised to 11 neurons are present in the human central nervous system. Supporting cells, which are called as glial cells, are 10 to 30 times more in number than the neural cells. The different parts of the neuron are a cell body, dendrites, and an axon that ends at the axon terminal. So this is the structure of neuron, showing cell, body, and the terminal. Okay, so the head is made up of dendrites, and it is occupied with the nucleus, where axon is myelinated with the myelin sheath by the Schwann cells, and these are the nerve endings with the terminal knobs. The cell body, which is also called as soma or perikaryon. The nucleus usually contains one nucleolus. Sometimes there may be two nucleoli. Centriole are absent, so the cell has lost the capacity to regenerate. Some contains genetic information and it is capable of protein synthesis. The soma at one end gives rise to small branching processes called dendrites, and at the other end a long process called as axon. The plasma membrane of soma is bilayered and lipid protein membrane called plasma lemma. Cytoplasm contains cell organelles, which includes Nissel's granules, mitochondria, Golgi apparatus, and lysosomes, and it also has a cytoskeletal proteins, which are neurofilaments, microtubules, and actin microfilaments. Nissel's granules, also called as Nissel's bodies, are stacks of rough endoplasmic reticulum with their membranes studded with ribosomes and polysomes. In Alzheimer's disease, neurofilaments form a characteristic neurofibrillary tangle. The dendrites are numerous short extensions from the cell body, they increase the cell surface area by many folds. Small knobby projections called as dendritic spines further increase the surface area, particularly in cerebral and cerebellar cortex. The dendrites receive the information from other cells and transmit to the cell body. They can cause protein synthesis, also generate and conduct the action potential. About 10,000 dendrites per neuron are seen in CNS. Axon, or the axis cylinder of the neuron, forms the nerve fiber, a long tubular process that extends away from the cell body to transmit output signals to the target organs. This arises from the thickened tapered area of the cell body, so which is called as axon hillock, or the initial segment of the axon. The action potential is generated at initial segment of the area in the motor neurons and the first node of Ranvier in the sensory neurons. Axoplasm contains mitochondria, Golgi apparatus, and cytoskeletal proteins. Axolemma is the plasma membrane of the axon. Different types of axons, they can be myelinated or unmyelinated axons. Myelinated axons have covering around their axons called myelin sheath. Myelin sheath is formed by Schwann cells in the peripheral nervous system and oligodendrites in the central nervous system. Myelin contains proteins, lipids, that is cholesterol and phospholipid and glycosphingolipids, and water. Large diameter of somatic nerve fibers as well as the preganglionic fibers of the autonomic nervous system are myelinated. Role of Schwann cell in myelination. The double layered of the membrane of a single Schwann cell wraps about around 100 times over 1 mm length of the axon, forming a covering of 8 to 10 micrometer thickness. The nucleus of the Schwann cell lies beneath its plasma membrane in the outermost layer. Thus, the myelin sheath of axon of a peripheral neuron is formed by many Schwann cells. Node of Ranvier. This is the gap between the Schwann cells, which is called as node of Ranvier, where the axonema is exposed to the extracellular fluid. Each node is 0.5 to 1 micrometer in length. The action potential travels from one node to another node, which is called as saltatory conduction, and thus conduction is faster in myelinated axon. The internodal distance is around 1 to 2 millimeters. The myelin sheath, the adjacent layer of the Schwann cell, stick to each other tightly. So with the help of a protein called as protein 0, which is present in the Schwann cell membrane. Mutation of this P0 causes defective myelination. In multiple sclerosis and autoimmune disease, patchy destruction of the myelin causes decreased conduction velocity in both motor and sensory neurons. Myelinogenesis. Axon invaginates into cytoplasm of adjacent Schwann cell. The axon remains suspended by a fold of Schwann cell membrane, which is called as mesoaxon. The mesoaxon becomes greatly elongated and spirally wrapped around the axon several times. The lipid gets deposited between the adjacent layer of the membrane. A thin layer of Schwann cell cytoplasm form an additional sheath called as neurolemma. The sensory fibers of the dorsal column system first get myelinated at 4th to 5th month of intrauterine life. The corticospinal tract fibers starts myelinating at 2 months of the age, and the process gets completed at about 2nd year of the life when the child is learned to walk. Axonal growth. At the growing end of the axon is present a growth cone, which is motile and contains actin. So the direction of the axonal growth is determined by the cell adhesion molecules and influenced by the trophic factors secreted from the target cells and guided by glial cells. So these are some of the functions of myelination. They increase the speed of conduction, it reduces the energy expenditure by the cell, it provides a protective covering to the axon, and it is also responsible for the color of the white matter to brain and spinal cord. Unmyelinated axons, they do not have myelin sheath. The somatic nerve fibers of small diameter postganglionic fibers of the autonomic nervous system, dorsal nerve roots, nerve fibers in the central nervous system, and the most of the fibers in the invertebrates are the example for unmyelinated axons. Speed of conduction of the impulse is slower compared to myelinated axons. Coming to exoplasmic transport, it means transfer of substances between cell body and axon terminal is called as exoplasmic transport. Various proteins, organelles, and other cellular substances required for development, growth, and maintenance of neuron are transported mainly along the length of the axon. They are abolished by application of colchicine, dinitrophenol, azide, cyanide, and prolonged anoxia. Colchicines disrupt the movement of microtubules, others block the process of oxidative phosphorylation. There are different types of exoplasmic transport, can be anterograde transport, retrograde transport, and transneuronal transport. So these are the exoplasmic transport, which is anterograde transport and retrograde transport. They are usually mediated by the microtubular system in the axon. Anterograde transport, they transport materials from the cell body towards the axon terminal. They can be mapped by H-leucine, for example, neurotransmitters. So there can be fast anterograde transport or there can be slow anterograde transport. Fast exoplasmic transport, the speed is around 400 millimeters per day. So they are mediated by the uh, microtubules kinesin, and molecules transported by anterograde transport are the cell organelles, vesicles, and membrane glycoproteins. And slow exoplasmic transport, where the speed is 0.5 to 2 millimeters per day, the material transported are actin, neurofilaments, and microtubules. Retrograde transport is where the transport of substances from axon terminal to cell body occurs. The speed is around 200 millimeters per day, and it is brought by dynein, and it can be mapped by Horsch-Radish peroxidase. Some of the examples of retrograde transport are transport of viruses. The chickenpox virus reaches the cell body from nerve terminals in the skin by retrograde transport. The rabies virus travels from the axonal ending of the motor neuron to the spinal cord and then brain to where it multiplies. The transport of toxins like tetanus toxin at the motor neuron end is transported to the cell body. The transfer of nerve growth factor, that is nerve growth factor, is taken up by the presynaptic terminal and transferred to the soma. The reuptake of synaptic neurotransmitter, the norepinephrine released at the nerve terminals, are rapidly removed from the synaptic cleft by reuptake into the presynaptic neurons. Transneural transport, where the trophic substances like nerve growth factors are transported across the synapse to the presynaptic membrane to another neuron. So this is called as transneuronal transport. So this helps in the maintenance of the synaptic contacts. Coming to the functions of neurons, the cell body, it maintains the functional and anatomical integrity of the axon. The synaptic transmitters are synthesized in the Nissel's granules of the cell body and are transported to the axon terminal by axoplasmic flow. Dendrites, they receive impulses and transmit the impulses towards the cell body. The axon hillock or initial segment, is the site where action potential is generated. The long axonal process propagates action potential from cell body to the axon terminal. The axon terminal contains the neurotransmitter in the vesicles. The arrival of action potential at the membrane of the axon terminal results in the release of these neurotransmitters. Special features of neuronal metabolism are the metabolism in the nerve for a considerable period is absence of oxygen. If oxygen supply is sufficient, the pyruvic acid is formed, and if oxygen supply is insufficient, the lactic acid accumulates. The energy requirement of the resting nerve is supplied primarily by the combustion of sugar and phospholipids. During activity, hydrolysis of ATP and creatine phosphate supplies the energy. Vitamin B12 is essential for oxidation of pyruvic acid and lactic acids in the neurons. Growth of neurons. Various factors that affect the neuronal development, growth, and the survival. So they can be categorized into the neurotrophins and other growth factors. In other growth factors, it can be ciliary neurotrophic factor, glial cell line derived neurotrophic factor, leukemia inhibitor factor, and FGF, PDGF, TGF, and IGF-1. Neurotrophins are the trophic proteins in the neurons as they promote the nerve growth and survival. They are produced by nerves, muscles, glands, and astrocytes. The known neurotrophins are nerve growth factor, which is called as NGF, and brain-derived neurotrophic factor, BDNF, neurotrophin 3, neurotrophin 4, and 5. Nerve growth factor is the first neurotrophin to be identified. It promotes the growth and survival of sympathetic nerve and some sensory nerves. The autocrine survival factor for a memory B lymphocytes. So it is found in high concentration in the saliva of male mice, and it also decreases the apoptosis of neurons by acting through tyrosine kinase A receptor. Brain-derived neurotrophic factor, it promotes the growth of peripheral sensory nerves and it acts through tyrosine kinase A pathway. Neurotrophin 3 promotes the growth of cutaneous mechanoreceptors and also it acts through tyrosine kinase A, B, and C. Neurotrophin 4 and 5 acts through tyrosine kinase B, and its exact function is not known. The types of neurons can be classified according to the number of processes, length of the axon, functions of the neurons, and pattern of dendrites. According to the arrangement of axon, they can be unipolar, pseudopolar, bipolar neurons, or multipolar neurons. Unipolar neurons have a single processes, usually found in invertebrate, in autonomic nervous system and cerebellum. Pseudopolar neurons, where axon after originating from soma splits into central and peripheral processes, for example dorsal root ganglia. Bipolar neurons have two processes arise from the body, example bipolar cells in the retina, ganglia of the vestibulocochlear nerve, olfactory receptor neuron. Then multipolar neurons, they have a many processes which arise from the soma, for example spinal motor neuron. According to the length of axon, it can be Golgi type 1 or Golgi type 2. Golgi type 1 have a very short axons. Their dendrites terminate near the soma, for example cortical inhibitory neurons. Type 2 Golgi neurons, their axons are long, for example cortical motor neurons. According to the function, they can be sensory neurons or motor neurons. Sensory neurons carry impulses from receptors to the central nervous system, whereas motor neurons carry impulses from the central nervous system to the target organs. According to the dendritic pattern, they can be pyramidal cells, where dendrites of these cells spread like pyramids, for example hippocampal pyramidal neurons. They're also called as stellate cells, where their dendritic pattern is radial shape spread of dendrites occurs in these cells, for example cortical stellate cells. So this is in brief about the structure and function of neuron. Thank you.